Hello everyone, my name is Bertolotti. I'm a program leader at the MRC Laboratory of Molecular Biology in Cambridge in the UK. And this is the second talk of a series of three that I'll be presenting to you. My first talk was giving you my personal perspective on the historical overview of protein phosphatases. And in this second talk, I will tell you how we discovered how we can selectively inhibit phosphatase, and there'll be a follow-up talk which will present for you a set of principles and assays that can be used to study phosphatases and identify selective phosphatase inhibitors. And in the background of all this, I'll try to illustrate for you the power and the benefit of phosphatase inhibition, particularly in the context of neurodegenerative diseases. So my lab has been interested in misfolded proteins for many years. These represent a huge problem for cells and organisms, and their accumulation in the form of insoluble aggregates is actually at the origin of a broad range of human diseases, including the devastating neurodegenerative diseases that affect an increasing number of individuals in our aging societies. So the disease shown here, Alzheimer's, Parkinson's, ALS or Lou Gehrig's disease, Huntington's disease, are all very different, right? They present with very different clinical manifestation. The faulty protein in each disease is different, but at the origin of these different diseases, I think what we have is a common molecular and cellular problem, which begins when cells fail to handle these proteins that then tend to misfold and aggregate, leading to catastrophic consequences for cells and organisms. Now, there's something very important, an important common feature to these diverse neurodegenerative diseases is the fact that they start late in life. And this is very important because it turns out that the disease-causing proteins are actually expressed throughout the life of individuals, but the diseases only start late in life. So what does it tell us? It tells us something very, very important. It tells us that we have mechanisms that normally function very well in neutralizing these bad proteins that we produce at all times. So what are they? These are sort of natural self-defense mechanisms against misfolded proteins. So we have, on one hand, chaperones that prevent protein aggregation. We also have degradation machineries, the proteasome and autophagy, that remove these bad proteins before they accumulate. And all these systems function very well together for years, for decades even, up to a point. As we age, it looks like the system gets overwhelmed, leading to accumulation of proteins that should normally be degraded. And this is really bad for cells and organisms because the accumulation of these proteins lead to a broad range of human diseases, such as the devastating neurodegenerative diseases. So if we appreciate that overwhelming natural defense mechanisms against misfolded proteins leads to a broad range of diseases, then a simple idea comes to mind, right? Our system normally works well for years, for decades even. So perhaps we can try and think about these self-defense mechanisms and try and boost them in order to correct the protein folding deficits that are causing a broad range of human diseases. So the idea is fairly rational, but then the big challenge comes, how on earth are we going to do this? So we've learned a great deal about protein quality control systems in the past 20, 30 years or so. And the only point I'm trying to make on this slide is these systems are very complex. We have multiple signaling pathways. They're all interconnected. We have negative feedback loops everywhere. So it's very difficult, although we've learned a lot about these mechanisms, it's very difficult to rationally predict how to improve the system for potential therapeutic benefit. And if you think about it, it's not too surprising because these systems have evolved through billion years of evolution. And what we want to do, we want to improve this system. So in other words, we want to beat Mother Nature and do better. So realizing this challenging, we decided to embark on unbiased approaches. By that I mean, rather than deciding, well, it should be good to modify things one way or the other, we let the cells tell us how we can manipulate them to improve their survival under catastrophic conditions that we in the lab create by blocking key components of protein quality control systems, leading to massive accumulation of misfolded proteins. So in this way, we sort of mimic the molecular problem which is at the origin of a broad range of human diseases. So I've sort of converted a complex organismal problem into a more tractable, if you like, cellular problem. And so by causing massive accumulation of misfolded proteins in cells, this usually kills cells. And this provides us with a really robust cellular model that we can use and ask, can we find ways to help cells survive in the presence of misfolded proteins? And our idea is, well, if we find a way to do that, perhaps this can lead to treatments that could benefit not just one, but actually a broad range of human diseases caused by accumulation of misfolded proteins. So over the years, in my lab, we've exploited this paradigm in many ways, but in today's talk, I don't have time to give you an overview of all what we've done. In some occasions, we've identified some approaches that actually led us to discover some new components of protein quality control systems. But in today's talk, what I would like to share with you is how we discovered an approach that helps cells survive massive accumulation of misfolded proteins, and how we've been able to implement this approach in models, in a model of a rare disease, and there's hope that perhaps approaches like this, aiming at helping cells surviving protein quality control or protein folding catastrophes, there's hope that we can perhaps develop these approaches for therapeutic benefit. So how did it all start? First, let me present to you the experimental system. So in the lab, we create artificially, we cause massive accumulation of misfolded proteins in cells, in this case by treating cells with a drug that is called tunicamycin. Tunicamycin blocks end-link glycosylation, and this is a post-translational modification which is important for proteins to fold in the endoplasmic reticulum, which is depicted here. And when we treat cells with tunicamycin, the proteins that are made in the endoplasmic reticulum, which, by the way, are proteins that will be destined to be secreted, such as hormones, or proteins that are destined to be at the cell surface, such as transmembrane receptors, those proteins are made in the ER. If we treat cells with tunicamycin, they can't fold, they accumulate in a misfolded state, and this is very bad. As you can see here, the cells just die under these conditions. So that provides us with a system that we can use, and we use this system in a sort of pro-survival screen to look for ways to help cells surviving under these catastrophic conditions. And we found a small molecule, which name is Guanabenz, which was fairly potent in protecting cells from this protein misfolding condition. By that I mean Guanabenz protected cells when used at concentration in the sub-micromolar range. So because it was fairly potent, and because we know rather well the mechanism by which cells defend themselves against accumulation of misfolded proteins in the endoplasmic reticulum. They do so by turning on a defense mechanism that is known as the unfolded protein response. I'd like to highlight the fact that this starts by a misfolding problem in the endoplasmic reticulum. So we know very well this defense mechanism, and it turns out that I contributed to discover some components of this pathway many years ago as a postdoc. So because our compound Guanabenz was fairly potent, and because we know well the mechanisms by which cells defend themselves against misfolded protein in the endoplasmic reticulum, we thought we would have a way to identify the mechanism of action of this cytoprotective molecule. And so we embarked on a detective expedition, as we like them, scrutinizing the different components of this cellular response. And the detective in this piece of work was a postdoc in the lab, Pavel Seitler, who found out how Guanabenz protected cells. And I'm now going to zoom into the middle branch of this pathway. So let me introduce this stress signaling event as an immediate first line of defense against many forms of stresses. Cells react by phosphorylating a translation initiation factor, EIF2 alpha, and as a result of that, this leads to a reduction of protein synthesis. And this is a signaling event which is vital for cells and organisms to cope with many forms of stresses. And an immediate benefit of this signaling event is as follows. Under normal circumstances, cells are busy producing thousands of proteins. In essence, a cell is a protein factory. And so if you decrease ever so slightly even the rates of protein synthesis, then this spares the availability of existing resources to handle the challenges. And this is how this phosphorylation of EIF2 alpha protects cells from many forms of insults. So that's for the first background information about this pathway. But it's also very important that this signaling event is transient, as many stress signaling events. Phosphorylation of EIF2 alpha needs to be transient because you don't want to slow down protein synthesis for too long. This would be detrimental. So this is why we, mammals, have evolved with two selective EIF2 alpha phosphatases, and these phosphatases belong to the family of PP1 phosphatases that I've introduced in the first talk. And these enzymes are peculiar in the sense that they are split enzymes. They are composed of two components that needs to be together for the enzyme to be fully functional. The one component is the catalytic subunit PP1, which turns out to be common to actually more than 200 holoenzymes. And the selectivity of the EIF2 alpha phosphatases is encoded by two non-catalytic subunits. The first one that was discovered is R15A, and it's not expressed in all cells. It's actually stress-inducible. R15A is made when EIF2 alpha is phosphorylated, and in this way, R15A is a component of a negative feedback loop that terminates this stress signaling event. But in addition to R15A, we also have R15B, which is functionally related to R15A, although different. It's encoded by a different gene, and it's actually expressed in all cells, as far as we can tell, and in all tissues. So if you read about this signaling event in the literature, what you will find is that this is a stress response. But the way I think about it today is actually more along the line of a rheostat, whereby phosphorylation of EIF2 alpha is controlled by the antagonistic action of the EIF2 alpha kinases and EIF2 alpha phosphatases to adjust the rate of protein synthesis to precisely match the demand. So that's it for the introduction on the signaling pathway. And this was to tell you that actually Guanabenz, remember this compound that protects cells from protein misfolding insults in the endoplasmic reticulum, does that by binding selectively to R15A, but it doesn't bind to R15B. And the consequence of that is Guanabenz inhibits R15A, and this actually prolongs the duration of the translation attenuation which results from stress. So stress leads to phosphorylation of EIF2 alpha, and as a result of that, protein synthesis is attenuated, and translation recovery is then mediated by R15A. Here we've discovered a selective inhibitor of R15A, which then acts by delaying translation recovery, right? And what's important to note is that although translation recovery in the presence of Guanabenz is delayed. It's not abolished, and this is because we have another phosphatase, R15B PP1, which is not inhibited by our compound. And this is very important. The selectivity of the compound here is key, because if we inhibit the two phosphatases, this is actually deleterious in the long run, because a persistent inhibition of protein synthesis is not desirable. Right? So to better show you the dynamic of this pathway, I've actually prepared a short animation that summarizes 10 years of work in 1 minute. So here we go. Under optimal circumstances, the cells are busy making thousands of proteins that are required to execute virtually all cellular function. And this process requires chaperones, this little hand in orange here, that are important in making sure that proteins fold properly and prevent aggregation. I've told you that we make errors, we make misfolded proteins to some degree at all times, but we are able to deal with our misfolded proteins. Chaperones are also important in targeting these abnormal proteins to degradation. Here I'm showing the proteasome. So this system works well up to a point. As we age or in diseases, we accumulate these bad proteins. And one way to defend ourselves against this consists in phosphorylating EIF2-alpha, slowing down the rates of protein synthesis. And this enables the chaperones that are normally busy in assisting the folding of newly synthesized proteins to actually deal with the misfolded ones. And this is one way this pathway is protective. So this led us to realize that we have here a very straightforward and very potent way to correct a protein folding problem. We can fine-tune protein synthesis by inhibiting R15A, and this in turn increases protein quality control capacity. So this is precisely what we were dreaming of doing, looking for strategies to treat protein misfolding diseases. And we were very keen to test this possibility. Unfortunately, as often in science, whilst these findings were really exciting, we couldn't quite go forward with Guanabenz. We had to take a big step back, which took some years. And the reason is as follows. It turns out that Guanabenz has another activity. It's not just the R15A inhibitor that I've described to you, but it actually was a marketed drug which was developed in the mid-80s for the treatment of hypertension. So for us, this is an undesirable effect because so Guanabenz is an alpha-2 adrenergic agonist. It has nanomolar affinity for its receptor. And so if we want to treat animals or mammals with R15, to cause R15A inhibition, the dominant effect of this molecule is actually to lower blood pressure. And this is a problem because it comes with coma and drowsiness at high doses. And actually, this compound is no longer used in the clinic. So when we read about the idea of drug repurposing, it's a tempting idea indeed, but we need to keep in mind that the first activity for which a drug might have been developed could become a big liability. And in this case, it is a big liability. We don't want to lower blood pressure. So we had to take a step back and look for selective R15A inhibitor. Fortunately, we realized very early on in the path that led to the discovery of Guanabenz, we realized that the activity of Guanabenz vis-a-vis R15A and protection from protein misfolding stress has nothing to do with the adrenergic receptor. The first reason for that is the cells we used in the experiments I've shown you don't express the adrenergic receptor. So that told us immediately that there was another target and that the two activities of this small molecule were separate. And so we reasoned that if the two activities are separate, we should be able to identify a compound that could selectively inhibit R15A, but not targeting the adrenergic receptor anymore. And so we initially, because Guanabenz had been developed in the mid-80s, there were a number of molecules closely related to Guanabenz that were actually commercially available. So we initially started with commercially available molecules, and we screened a number of such molecules. And I'm sparing you the failures here, but most of the molecules we screened were completely inactive against R15A. But one of them remained active, and we call this molecule cephin-1. And as you can see, it's a very close derivative of Guanabenz. It's only a small modification. So here we've removed the chlorine substitution in position 6, and you'll see this dramatically changes the property of this small molecule. But first, let me show you that cephin-1, as Guanabenz, binds selectively to R15A, and as a result of that, it protects cells from this protein misfolding in cells that we trigger with tunicamycin. And importantly, we find that if we use cells that are knocked out for R15A, we lose the cytoprotective effect of cephin, which suggests that all the measurable effect of the compound that we see here at these concentrations are due to inhibition of R15A. So we are looking at an on-target effect of this molecule. Our good fortune here is that cephin is closely related to Guanabenz, and Guanabenz had been initially developed as a centrally active drug, so it crosses the blood-brain barrier, and so does cephin. Both are orally available, rapidly distributed to tissues, and we performed some tolerability studies in mice initially and found that cephin is well tolerated, and we didn't observe any overt toxicity. So this was exciting, and it was really important indeed to develop cephin. Here I'm showing you an experiment where you can see mice that had been treated with Guanabenz from 1 or 10 milligram kilo, doses that we need to inhibit R15A in vivo. You see that we have massive sedation. The mice don't move actually, and this is due to the adrenergic activity of Guanabenz. But the cephin-treated mice, as you can see, don't have this problem because the removal of this substitution on cephin has engineered out the adrenergic activity. So this was great. We have a compound. We can work out the concentration we want to use it if we want to use it in vivo. So the way this worked was a combination of pharmacokinetic studies where we measure the concentration of the compound in the tissues of interest combined with the potency of the compound as we've seen in cells. And so we've chosen to treat mammals with a concentration ranging from 1 to 5 milligram per kilo. This corresponds to the concentration used here in cells where we know the compound is selective. So great, we have a small molecule, it took us many years to get there. We have a small molecule that we can use to test this initial idea that was in the back of our mind for a long time. Can we improve protein folding in mammals, and can we use this approach to treat neurodegenerative diseases? And don't get your hopes too high, because the disease I'm going to discuss now is none of these common neurodegenerative diseases. We actually turned our attention to a rare disease, and the reason is because of the way cephin and Guanabenz were discovered. So remember, we found that both cephin and Guanabenz protected cells from protein misfolding problems in the endoplasmic reticulum, right? This is a specific subcellular localization, whilst the other neurodegenerative diseases, the common neurodegenerative diseases shown here, in this disease, the misfolding pathology is not in the endoplasmic reticulum, but actually in the cytosol or in the nucleus. So realizing this, we thought, let's be careful here and look for a disease where the misfolding pathology is in the endoplasmic reticulum. And so that got us interested in Charcot-Marie-Tooth 1B. It's a very rare peripheral neuropathy, but it's a clear, what I'd call ER stress disorder. So this disease is caused by a dominantly inherited mutation in a protein called myelin protein 0, which is one of the most abundant proteins that is made by Schwann cells in our peripheral nervous system. And as a result of the CMT1B causing mutation, the protein misfolds, and actually it is, for this reason, retained in the ER as a misfolded protein, and this causes a typical ER stress response. This has been elucidated largely by Larry Rabbit together with Maurizio D'Antonio. So we teamed up with Larry and Maurizio, who provided us mice transgenic for myelin protein 0 with the mutation causing a form of Charcot-Marie-Tooth 1B. And we characterized these mice as they had seen before us, and we found that as in human, the CMT1B mice display motor deficit with time. If we ask them to run on a rata rod, they don't perform very well. They tend to fall off, actually. And this is because their Schwann cells is exhausted due to the accumulation of massive amounts of the misfolded proteins causing Charcot-Marie-Tooth 1B. And as a result, there's a very thin myelin layers along the axons of these nerves. It's a very obvious deficit. So a really talented postdoc in my lab, Indrajit Das, went on and treated these mice for 5 months by oral gavage. It was a big effort, but it did pay off because he found that cephin actually prevented the motor deficit in these transgenic mice. And this came about because it rescued myelin thickness in the sciatic nerves of these mice. And this is because it alleviated the stress resulting from the misfolding of these proteins. So following the work I've described to you with both Guanabenz and cephin, a number of clinical trials have been started. First with Guanabenz because it's an FDA-approved drug, but some obviously encounter the same problem as I've shown you with mice. The adrenergic activities is a problem, and there's recently a trial that has started with cephin. This is exciting, but it's obviously too early to tell whether it could be applicable in human. But surely we've learned through this work that we can enhance in this way protein quality control, not only in cells but also in a mammal, and this can have a broad range of therapeutic benefits. So this is exciting, particularly for me, who have spent now 20 years studying the way cells respond to tunicamycin stress. And I must say, when we started this 20 years ago, this was thought to be esoteric, boring piece of work. You know, who cares how HeLa cells deal with tunicamycin? Well, it turns out that tunicamycin was a probe that enabled us to identify components of a signaling pathway which is evolutionarily conserved and therefore important. We had no idea whatsoever that there could be a potential therapeutic benefit to manipulating this pathway. But the point I'm trying to make here is that because a cell is a basic unit of an organism, whatever we study will inevitably have some medical or therapeutic relevance one day or another. So it is worthwhile to continue studying the unknown. So I want to close on a serendipitous discovery that came with this work, which was the realization that we can selectively inhibit a phosphatase by targeting its regulatory subunit. And this was good news because phosphatases had acquired a very bad reputation over the years. They were thought to be undruggable. So this started because people started generating catalytic inhibitors of PP1, which is the common catalytic subunit to about hundreds of phosphatases in the cell. So if you inhibit PP1, it's not one enzyme that is inhibited, but actually hundreds, and that's toxic. But here we found, as I presented to you, a way to selectively inhibit a phosphatase by targeting its regulatory subunit. So we thought, well, in principle, we could perhaps consider inhibiting the other phosphatases in the same way, and in this way open up a broad range perhaps of ways to manipulate cell function, perhaps for therapeutic benefits. Remember, phosphatases control virtually all biological processes, so there's a great opportunity here to tap to inhibit a class of enzyme that had been previously untapped. And this is exciting, but we had to find a way to design methods not only to study these phosphatases but also to identify rational inhibitors. And this will be a topic that I will develop in my third presentation. So with that, I'd like to thank you for your attention and thank also all the people from my lab who participated in this work. These are all my lab members. May look like a small group, and that's a typical feature of LMB. We have small groups, but very talented people who did the work I just presented to you. Thank you very much.